This is a quick general overview of cellular structures. Alright, so we are going to begin with the levels of structural organization, starting with atoms. Atoms are the basic building blocks of matter. They are the smallest level of structural organization. Groups of atoms that are bonded together are molecules, and molecules are the smallest fundamental unit of a chemical compound. Molecules that are bonded together to create a very large molecule is a macromolecule. And some examples of macromolecules are proteins, lipids, carbohydrates, and nucleic acids. And so these groups of macromolecules function together to be a cell, right? And a cell is the basic unit of life. Groups of cells that function together form tissues, and groups of tissues that function together form organs, like your liver. Groups of organs that function together are then known as organ systems, and there are 11 organ systems, and if you take those organ systems as a whole, you then are looking at the organismal level of structural organization. So this particular video is regarding the cell, right? So the cell is the fundamental unit of life. Cells are really interesting. They create their own energy, and they are also self-replicating. There are also many different types of cells, each with their very own function. You have a blood cell that transports oxygen, a sperm cell, which is on its way to fuse with a gamete, with an egg cell, to create a zygote. And then there's the egg cell, which is the female gamete, and the nerve cell, or the neuron. Organelles are specialized structures that are characteristics shared across cells. They include the Golgi apparatus, centrosomes, the cytoplasm, ribosomes, lysosomes, the smooth endoplasmic reticulum, the rough endoplasmic reticulum, the mitochondria, plasma membrane, vesicles, the nucleolus, and the nucleus. So we're going to begin with the plasma membrane. The plasma membrane is the outer surface of the cell. It is comprised of a phospholipid bilayer. And these lipid molecules that are forming the phospholipid bilayer are amphipathic, meaning that they have both a hydrophilic head and a hydrophobic tail. Hydrophilic meaning water-loving, hydrophobic meaning water-fearing. And this hydrophilic head gives this plasma membrane a polar characteristic, meaning that the water-soluble molecules are attracted to the hydrophilic heads. So the hydrophilic heads are on the outside of this bilayer, and they're interacting with the cytoplasm inside of the cell and the extracellular fluid on the outside of the cell. On the inside of the phospholipid bilayer are the hydrophobic tails. And hydrophobic, water-fearing, right? So water-soluble molecules cannot pass directly through this membrane. So any water-soluble molecule has to use proteins for cross-membrane transport. So you can see in the picture, there are lots of transport proteins. There are also integral proteins, peripheral proteins, surface proteins, all sorts of proteins that are going to aid in the passage of molecules that are molecules that are water-soluble through the plasma membrane. And in essence, what this is doing is controlling the cell's interactions with the outside environment. The centrosome is an organelle that contains the centrioles from which those spindle fibers develop during mitosis and meiosis. A centriole, in essence, is just a small set of microtubules that attach to the mitotic spindle. And these centrioles are generally found in pairs at opposite ends of the nucleus. Alright, next is the cytosol. The cytosol is just fluid that's found inside of living cells. It's a water-based fluid, so it contains water-soluble molecules, and thus it forms an electrostatic charge with those hydrophilic or water-loving heads of the phospholipid bilayer. Now, a lot of people confuse the cytosol with the cytoplasm, but they're not necessarily the same thing. The cytoplasm, while it consists of cytosol, it also consists of organelles and carbohydrates and lipids, so things that are insoluble in water. The cytoplasm, in essence, contains the total content of the cell membrane, other than the nucleus, which the nucleus is separated by its own nuclear membrane. The ribosome is a particle located in the cytoplasm that serves as the site of protein synthesis. So this is where the translation of messenger RNA to form a protein takes place. Ribosomes can be free particles, or they can be attached to the rough endoplasmic reticulum. Ribosomes, in essence, are made of ribosomal RNA, and they are composed of two subunits, large and small subunits. You can see in this picture, the mRNA is being read, and the tRNA, you see that growing polypeptide chain of amino acids, which are proteins coming off the back. The smooth endoplasmic reticulum and the endoplasmic reticulum in general, they're a system of interconnected tubules and cisternae, which are just flattened sac-like structures. Interestingly, the endoplasmic reticulum is not found in blood cells. Now, the way to distinguish the smooth from the rough ER is that the smooth ER does not have ribosomes attached to its surface, and the smooth ER functions in the production and metabolism of fats and steroids, such as cholesterol, and it also detoxifies harmful metabolic byproducts. The rough endoplasmic reticulum, on the other hand, ribosomes are attached to its surface, and therefore this is where the synthesis and transportation of proteins occurs. Now, a lysosome, this might be one that not many have heard of before. A lysosome is this complex of enzymes. It contains degradative hydrolytic enzymes, and hydrolysis is just a chemical process that causes a molecule to split into two separate parts after an addition of a water molecule. So this is how macromolecules are digested, through hydrolysis. These enzymes are enclosed within a membrane, and this membrane is meant to protect the cell from any leakage from the lysosome, because the lysosome environment on the inside, or the interior, is very acidic, but the pH of a cell is fairly neutral, if not alkaline, so it needs to protect the cell from any acidic leakage of the lysosomal interior environment. The Golgi apparatus is a flattened disc-like structure that's located near the ER. This is where proteins and lipids are received. They're sorted and then packaged into vesicles, and these vesicles can go to a lysosome to be digested. They can go into and out of the plasma membrane outside of the cell. There are three parts to the Golgi apparatus you're going to want to know. The cis network, that's where everything enters. It's the receiving side of the Golgi apparatus. The Golgi stack is where everything is processed, and the shipping side of the Golgi apparatus, where everything is sorted and released, is the trans network of the Golgi apparatus. The mitochondria is the powerhouse of the cell. This is where oxygen is used to create energy in a process known as cellular respiration. The mitochondrial matrix is the location of the citric acid or Krebs cycle, and the inner mitochondrial membrane is the location of the electron transport chain. And the final step in cellular respiration along that electron transport chain is oxygen as the last electron acceptor. And this creates an abundance of ATP, and ATP is energy for cells. Something interesting about the mitochondria is that it is thought to have originated from a process known as endosymbiosis, which is where a cell engulfs, or they believe that a cell engulfed an aerobic prokaryote, and eventually they formed a mutualism with one another, and that is how mitochondria came to be. Alright, the nucleus is very important. The nucleus regulates activities of the cell. It is the location of DNA or hereditary information. It also has what are called nucleoli, that are just small particles within the nucleus that function in RNA synthesis. The gel matrix inside the nucleus is called the nucleoplasm. And if you remember, during mitosis and meiosis, the nucleus actually, or the nuclear membrane, breaks down during cellular division. Now, the nucleolus is this dense sphere in the middle of the nucleus of the cell, and this is where ribosome biogenesis and assembly takes place. And so you may have noticed that cells have an abundance of ribosomes because they're constantly making proteins, and proteins are what are needed for any physiological process to take place. Alright, And last but not least, the cytoskeleton. The cytoskeleton is just a network of filaments. There are about 3 types of protein fibers that make up the cytoskeleton. There are microtubules that support the cell and give it its shape. The microtubules provide tracks for motor proteins called connensins and dyneins, which transport vesicles. The intermediate filaments, they strengthen the cell and they help maintain the shape. They also aid in positioning of the organelles. Keratin, for instance, forms intermediate filaments. They are found, keratin, is found in hair, skin, and nails. It's basically a filament that is specialized to bear tension. Microfilaments are the 3rd protein fiber. They anchor the cytoplasm to an integral protein of the plasma membrane, and that enables cellular movement. So these microfilaments are composed of actin monomers, and so they're also sometimes called actin filaments. And these actin monomers enable cellular movement by interactions with a motor protein called myosin. Alright, so that's all the organelles found in a cell that you guys will have to remember. Their functions, you should be able to identify them if given a model. And I really hope that this helps you guys study, so we'll see you next time.